بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. Hi everyone. The last lecture was about the first disorder of iron metabolism, which was the iron deficiency anemia. Today's lecture will be about the second and the opposite disorders, iron overload or hemochromatosis, which is more and more serious than the iron deficiency. If we want to define hemochromatosis, we will say that it is an abnormal accumulation of iron in the parenchymal organs. What does the mean of that? Let us remember that iron storage may take either the form of ferritin or hemosiderin. Do you remember when we said that ferritin is an iron core enveloped in a thick cortex of protein, which is enough to protect tissue from iron toxicity. So, ferritin, when accumulates in tissues, it doesn't cause harm. On the other hand, hemosiderin is an iron with a much smaller amount of protein around it, which is not enough to protect tissues from iron toxicity. So, hemosiderin is allowed only in the normal circumstances to accumulate in the reticuloendothelial tissues, which can manipulate iron toxicity without harm. In the parenchymal organs, when hemosiderin accumulates due to iron overload, it will cause harm and an iron toxicity to the affected organs. In iron overload, iron will accumulate in the form of ferritin till it exceeds the capacity of ferritin. Then it accumulates in the form of hemosiderin in the reticuloendothelial tissues till it exceeds the capacity of these tissues. Then it begins to accumulate in parenchymal tissues and causes harm and organ toxicity. How come? We said before that iron is a tightly regulated element, and we had illustrated the mechanism of tight iron regulation by Hebstein, which will certainly prevent iron overload in normal circumstances. What can cause iron to increase and hemosiderin to accumulate in parenchymal organs? Certainly, this occurs when there is a defect in the mechanism of iron regulation by Hebstein, or when iron enters the circulation by process beyond the ability of Hebstein to control. First, you must refer to the lecture of iron metabolism to remember the mechanism by which the hepatic cells sense the excess iron, and then a number of molecules and receptors are stimulated on the surface of the hepatocyte, followed by transmission of signals to the nucleus of hepatocytes and stimulation of the promoter of hemp gene to produce hepcidin. Any defect in this pathway will affect the induction of hepcidin secretion by the excess iron, and thus the negative feedback inhibition of iron accumulation or iron entry in the circulation from either the reticuloendothelial tissues or the GIT. Also, the iron may enter the circulation by another way, which is beyond the control of Hebstein. For example, through intravenous iron intake or through blood transfusion. Do you know what is the meaning of ineffective erythropoiesis? Ineffective erythropoiesis means an intramedullary destruction of RBCs and their precursors, or destruction of RBCs and their precursors in the bone marrow. We had illustrated how does the ineffective erythropoiesis affect hepcidin secretion through release of some mediators from the erythroblast during erythropoiesis, as growth differentiation factor 15 and twisted gracilation 1, which inhibit hepcidin. Patients suffering from the ineffective erythropoiesis as thalassaemic patient will have iron overload, and one of the mechanisms involved in that is the effect of ineffective erythropoiesis on hepcidin. Other mechanisms include frequent blood transfusion and hemolysis, which increases the recycling of iron from the destroyed RBCs. Other causes of iron overload include a defect in the ferroportin known as sub-Saharan African dietary iron overload. These patients have a defect in the ferroportin which makes it irresponsive to hepcidin. The entry of iron from GIT into the circulation will increase in these patients. Also, in this area, food containers are instructed with iron, which make escape into food and increase the iron content in diet. This also can result from chronic oral iron intake of iron when combined by defect in the ferroportin. Iron overload may be focal as a pulmonary hemosiderosis resulted from pulmonary hemorrhage, chronic hemoglobinuria, which causes iron overload in the renal tubules and kidney, mitochondrial iron overload causing ataxia, and a hereditary disorder of hyperferritinemia, which causes iron overload in the eyelids causing cataract. Rare causes of iron overload include congenital transferrinemia, congenital atransferrinemia, or congenital aceruloplasminemia, which causes iron overload in organs. Now we will understand the mechanism of organ toxicity by iron. Again, you must refer to the lecture of iron metabolism, where we said that iron can be present in two forms, ferrous and ferric form, and that is easy to interconvert between the two forms through the redox reaction, which release free oxygen radicals. These free radicals causes peroxidation of the lipids of the cell membrane and membrane injury, oxidation of the proteins inside the cells, injury of the lysosomal membrane and release of lysosomal enzymes, which causes autolysis of cells. This is how the iron overload causes iron toxicity and organ toxicity. Please return to this diagram in the lecture of iron metabolism to remember the mechanism of hepcidin regulation by iron or hepcidin induction by iron. After that, we can say that the defect in any protein demonstrated in this pathway can cause iron overload. On this base, hereditary hemochromatosis had been divided into 4 types, from which the most common is the type 1 or HFE type, to the extent that some schools had divided hemochromatosis into HFE and non-HFE. Type 2 is caused by defects either in the hemojuvenile or in the hemp gene itself. Type 3 is caused by defect in the transferrin receptor 2, and type 4 is caused by defect in the ferroportin. The first 3 types are more serious than type 4, and the first 3 types are autosomal recessive, while the type 4 is autosomal dominant. Here is the first type, or the HFE type. It's caused by an autosomal recessive mutation in the HFE gene on chromosome 6, mostly by C282Y or H63D mutations. The C282Y means substitution of the normal amino acid cysteine at the position of 282 of amino acids by tyrosine. The H63D means substitution of the normal amino acid histidine at the 63rd position of the amino acids by aspartate. These mutations cause dysfunction of the HFE protein, which makes the hemp gene irresponsive to the iron excess. So, iron exit from macrophages and iron absorption from GIT is not inhibited by hepcidin, causing iron overload and toxicity of parenchymal organs. Clinically, this type is manifested by cardiac manifestations, hepatic manifestations, endocrine manifestations, and the bronzed color of the skin. This is the cause of naming this type bronzed diabetes. There is also arthritis and joint manifestations. Type 2 is called the juvenile hemochromatosis, as it is presented earlier than type 1. It is usually presented at the second or third decades. It is caused by mutation of either homojuvenile gene on chromosome number 1 or hemp gene at chromosome number 19. This mutation causes irresponsiveness of hepcidin to iron excess and iron overload. It is presented by cardiac manifestations and hypogonadism. Type 3 is caused by an autosomal recessive mutation of the transferrin receptor 2 gene on chromosome number 3. This mutation causes irresponsiveness of hepcidin to iron excess and iron overload. It is presented by clinical picture similar to but more severe than type 1. All these three types causes parenchymal iron overload and organ toxicity. Type 4 is rare and is caused by autosomal dominant mutations in the ferroportin gene on chromosome number 2. It is either causes a decreased ferroportin expression, decreasing the iron exit from macrophages, hepatocytes, and GIT cells. This causes iron deposition in the reticuloendothelial system, which can manipulate with iron and causes no harm to tissues. Either mutation of ferroportin makes ferroportin irresponsive to hepcidin, increasing iron exit from macrophages, hepatocytes, and GIT cells, increasing the iron in the circulation and causing iron deposition in the parenchymal organs and organ toxicity as the first three types. Now, we will talk about the lab diagnosis of iron overload, which includes proving that there is an iron overload, diagnosis of tissue damage caused by iron overload, and diagnosis of the cause of iron overload. Detection of the iron overload can be done first by iron profile, which will demonstrate an increase of serum iron, increase of serum ferritin, increase of serum transferrin saturation, and decrease of total iron binding capacity. The most sensitive of these is the transferrin saturation. Iron overload is diagnosed when the transferrin saturation is more than 55% in males and more than 50% in females. Iron stores can be measured by tissue biopsies as liver biopsy, bone marrow biopsy, and skin biopsy. Liver biopsy had been replaced by the MRI liver. Iron stain of bone marrow will demonstrate an increase of the percent of sideroblasts, an increase of iron in both macrophages and sideroblasts, and random distribution of hemosiderin granules in the cytoplasm of sideroblasts. Gleason test, which is an obsolete test, will demonstrate more than 2 mg iron in 24 hours urine. Phlebotomy test, which is also an obsolete test, will demonstrate excessive venous sections required till iron depletion occurs. For more understanding of these tests, please refer to the lecture of iron investigations. The second step is detection of tissue damage caused by iron overload. This can be detected by organ function and measurement of serum levels of hormones, or radiological tests on bone and heart and ECG. The third step is detection of the cause of iron overload by genotyping. Genotyping by PCR will give a definitive diagnosis. If the patient is positive for any of the mutations causing hemochromatosis, family members should be screened by transferrin saturation test and the serum ferritin test, and if these were high, genotyping is recommended. Findings in type 4 is different, as the iron deposition will be more in the reticuloendothelial system and not in the parenchymal organs. Serum ferritin will be high but with normal transferrin saturation, as the iron is restricted in the reticuloendothelial system. Treatment includes regular phlebotomy until the transferrin saturation is less than 50% and serum ferritin is less than 50 microgram per liter. The second method is using iron chelator, which chelates the excess iron and is excreted in urine. I hope the data in this lecture was clear and that you had understood the iron overload. See you soon. God bless you. والسلام عليكم ورحمة الله وبركاته.